All right. Today in AP Bio, we're going to talk about the structure and function of the cell membrane. Now, the cell membrane has a super important job. So important that it's going to be central not only in the rest of this unit when we talk about cell transport later on, but also next unit when we talk about cell communication in the cell cycle. Here is a diagram, a picture up close of the cell membrane. And there's a lot of stuff going on that we're going to look at. So firstly, and most importantly, we should remember from our first unit that cell membrane is composed primarily of phospholipids. Now here's two pictures on the right side of a phospholipid. And remember, a phospholipid is composed with a glycerol molecule and two fatty acids attached to it, along with a charged phosphate group. Because phospholipids are amphipathic, meaning they're part polar and nonpolar, they contain a phosphate head which is hydrophilic because it's polar, and the fatty acid nonpolar tails that are hydrophobic. Because of this, they form a bilayer, which you see on the bottom right here. So the phosphate heads that are polar, that are hydrophilic, are facing the intracellular and extracellular fluid. That leaves the hydrophobic nonpolar tails scrunched together in the middle to avoid and repel water. Here's a picture of them arranged in a bilayer, and it labels the polar regions and the nonpolar regions. Now, one of the central parts of this whole lesson is relating how the structure of the cell membrane relates to the things that are able to freely cross it and those that can't. Okay, so charged substances, polar substances, and large molecules are not able to freely cross the plasma membrane. Charged substances and polar substances can't cross the cell membrane because they do not mix. They're not able to coexist in the nonpolar tail sections of the plasma membrane. They don't mix with it, so they can't cross this nonpolar tail section. Large molecules can't because think about like if you I'm a big fan of concerts, right? When I was growing up and still like I go to concerts all the time like every weekend if I could. But right at the beginning of a concert, if you want to be way up to the front, you might have space standing around waiting for the performers to come out on stage. But when they do, when the lights all of a sudden dim and then they're coming out, everybody squishes towards the front. And you might have somebody trying to run towards the front at the end and, right before the show's going to start. And sometimes you get these really big guys trying to get up towards the front, and they're just too big. They can't make it through. Everybody else stuck shoulder to shoulder. It's the small, sneaky people, real short that can weave in between everybody to get to the front. It's the same thing with the plasma membrane. Large molecules just take up too much space. They can't push their way through, even if these large molecules are nonpolar, they can't push their way through the plasma membrane. So the plasma membrane is impermeable to large substances also. So in general, what molecules can get through the plasma membrane easily? Small nonpolar substances. So small fats and small lipids can get through. And also small uncharged substances like uh, oxygen or carbon dioxide. The things that can't get through are polar molecules. Like even water has a hard time getting through. That's why the cell membrane has to have in some places where water needs to get through relatively easily, these channels called aquaporons. Ions can't get through, and large molecules, even if they're nonpolar, can't get through very easily. The plasma membrane also behaves very differently depending on the predominant types of fatty acids that are part of the phospholipids. So membranes must be fluid and flexible. In general, they're about as fluid as thick salad oil. Now, the percentage of unsaturated fatty acids that are in the phospholipids determine kind of in part how flexible they are. So if a plasma membrane has more unsaturated fatty acids as part of their phospholipids, the membrane is less viscous. That means it's less thick and it's more fluid. You will find this is especially the case in cold adapted organisms like winter wheat or Arctic salmon. Um, and in some organisms, like winter wheat specifically, the composition of their cell's plasma membrane will change towards autumn. The percentage of unsaturated fatty acids will actually increase in the autumn months in preparation for the cold. Because when it gets cold, everything moves slower, and if we do not keep it fluid enough, it'll be very hard for substances to move in and out. In addition, cholesterol molecules, and that's what these yellow pieces are in this diagram here, also help the membrane stay more fluid by taking up space and spreading out the phospholipids. So we talked about how cholesterol has many important roles, and one as a component of the membrane to help maintain its stability and fluidity. The cell membrane defines the cell. It separates the living cell from its aqueous environment, and it's only about 8 nanometers thick. Only about 8 nanometers or 8 millionths of a meter thick. It controls all the traffic in around and out of the cell, and it allows some substances to cross more easily, like non-polar or small molecules than others. We call this semi-permeable. Permeable, able to permeate or cross it. Some things are, and some things aren't. Now, you might think, okay, well, so since large or polar or charged molecules can't cross easily, how do things like uh, steroids, or how do things like ions, like salt, or how do things like monosaccharides that the cell needs to move in and out of the cell cross then if they can't just go through the plasma membrane? And the answer is that the cell has channels or little doorways made out of protein to help those substances that couldn't normally cross the membrane in and out. And these channels are usually very specific to the items or the substances that they're allowing to cross. So there might be a glucose channel, and there might be a water channel, and there might be a channel for amino acids, and all of them are different to only let certain things through one specific channel. Here is a picture on the right of what that protein channel might look like a little bit more realistically than these other models here on the left. Now, the cell membrane is much more than just phospholipids and cholesterol. It contains transmembrane proteins embedded in the phospholipid bilayer. So here on the right, here are transmembrane proteins. They're called transmembrane because they span the whole membrane. There's part of it sticking out and there's part of it sticking in. Or they're also peripheral proteins. Look at these proteins that are on the periphery. One edge, the extracellular edge, or the intracellular edge only. Now, you might be asking, why are proteins the perfect molecule to build structure in the cell membrane? Why use proteins as channels instead of other molecules? And the answer is, remember how proteins are made up of amino acids that have different properties. Like some, like this, are non-polar amino acids that are hydrophobic, and others are polar amino acids that are hydrophilic. That difference in amino acid property makes proteins the perfect substance to act as channels and integrals in the membrane. So within a protein or within a membrane, not sections of the protein might be composed of different parts that have more or fewer polar or non-polar amino acids. So sections of a protein that are composed of mostly non-polar amino acids that are hydrophobic would be very good to be part of the protein that's found in the nonpolar region of the phospholipid bilayer. They usually serve as anchors, keeping the protein in the phospholipid bilayer, because if you try to push the protein one way or the other, those nonpolar amino acids would be exposed to water, which they repel, they don't like. And the outer surfaces, this part that's in purple, and the extracellular intracellular fluid of a protein channel are mainly made of polar amino acids that mix well, that are attracted to are hydrophilic, and they extend into the both domains. So the fact that these proteins can be made of amino acids in segments that have a greater concentration of polar versus non-polar amino acids make them the perfect things to make up to be spanning the phospholipid bilayer. Now, here's some different examples of protein channels. Aquaporins are water-specific channels in bacteria and other type of cells that facilitate the movement of water into and out of the cell. Now, water is a very small molecule, but it's also very polar, so it sometimes can be difficult for it to move in and out of the cell in the quantity and rate that we would like. Here is a proton pump, which moves an H plus, a positively charged hydrogen ion, into and out of the cell. Right? These protein channels function by a conformational change or shape change. They are normally, let's say, closed or prevent things from passing. And when the correct molecule, water or a proton, binds with them, they change their shape to push and move that substance into or out of the cell. Now, these membrane proteins can have many different functions. They can work as transporters, they can work as enzymes, they can work as cell surface receptors to transmit messages or help to identify uh, a cell from as a hey, I'm a good guy, I'm a body cell versus hey, I'm a bad guy, I'm a pathogen. They can help cells stick to each other, and they're also useful in anchoring the cytoskeleton that gives the cell its structure. The proteins in a cell's membrane help with specific functions. So cell membranes and organelle membranes each have unique collections of proteins. We talked about peripheral proteins that are loosely bound to the membrane surface and integral proteins that span the membrane. In fact, in 1972, S.J. Singer and G. Nicholson proposed that membrane proteins are inserted in the phospholipid bilayer, like we talked about, and they proposed what's called the fluid mosaic model, or the fact that the cell membrane is kind of like a loose fluid. The phospholipids kind of flow, and they have uh, proteins, the mosaic stones that are stuck in there to kind of give it a more rigid structure. So it's like a fluid, it's like a mosaic, it's a fluid mosaic. Here is a picture of the whole cell membrane, and you'll see some common features. You'll see the phospholipids and the cholesterol molecules. You'll see membrane proteins, some transmembrane proteins, and some proliferal proteins. You'll also see what's called glycoproteins. And there are proteins that have carbohydrates attached. So glyco, like glucose, means uh, carbohydrate. And you will see those have the string of carbohydrates attached to this protein. They're really important for cell-to-cell recognition. And you'll also see glycolipids. So in place of the phospholipid head, they have monosaccharides or polysaccharides, and those are helpful for cell signaling as well. You'll also see on the inside of the cell membrane is a place where the cytoskeleton can integrate and attach to the phospholipid bilayer to give it the cell structure. So here's another picture of a cell carbohydrate, like a glycoprotein, and they play very important roles in cell recognition and the ability for one cell to distinguish itself from another. And they're important in organ and tissue development, and they're really the basis for rejection of foreign cells by the immune system. So they act like as tags to say, hey, this cell, this is part of our body, we're all good. Now, I hope you enjoyed this section that's talking about the plasma membrane because it's super important for what we're going to talk about next.